All right, this is being recorded, Ed, is that right?
That is
I hope
correct.
so. Otherwise, I'm gonna get in trouble from my parents.
Nope, you're
No one
good.
I fear more than my mother.
Not here.
Hmm.
Relax.
Oh, she's here. Thank you, Nadia, and thank you everyone for being here. I really appreciate it. It's a very special day for me. So as many of you know, aging is a leading risk factor for many diseases, particularly age-related cognitive decline and also Alzheimer's disease dementia. And we know from heritability studies that genetic makeup plays an important role in determining how an individual ages, whether it's this successful trajectory with little cognitive decline, a more normal trajectory with some cognitive decline, or this, a severe descent into dementia, which is when cognitive impairment becomes so severe, it interferes with daily living abilities. And we think that identifying the genes and pathways involved in regulating an individual's trajectory will not only enable a better understanding of the causes of age-related cognitive decline and Alzheimer's disease dementia, but also identify potential therapeutics. And so here I'm showing you a conceptual framework of how these individual differences in cognitive decline might emerge. And don't worry, I'm going to break it down for you. So we're going to start with normal, non-pathological cognitive aging. And in this example, there are molecular and cellular, as well as global brain changes that correspond to individual differences in cognitive decline, albeit they're occurring in the absence of classic brain neuropathologies that are used to definitively diagnose Alzheimer's disease. And these are amyloid plaques and neurofibrillary tangles. And so it's thought that individual differences in normal, non-pathological cognitive decline arise by virtue of both genetics and cumulative environmental exposures across the lifespan, with things like early life cognitive enrichment, physical exercise, and diet being considered as protective. And some of this is covered in more detail in our recent publications on environmental factors that I'm not going to have time to discuss much today. Regarding the genetic component, I want to briefly highlight our collaborative work with the Chesler Lab, where we were looking for specific genes that control individual differences in normal, non-pathological cognitive decline using the diversity outbred mouse population as our model system. And so just to remind everyone, the DO mice are a population of outbred mice that are segregating approximately 50 million polymorphisms. So they model the genetic diversity of humans. And here I'm going to show you that we use them to identify genetic variants associated with working memory decline using ultra-high precision mapping. So first, Laura Anderson measured working memory in a large cohort of DO mice using a cross-sectional design as part of the shock center phenotyping core. Next, Andrew Willett and Naran Haddad analyzed these data and found that working memory declines with age across the DO population. And then they performed genetic mapping and included age as an interactive covariate. And using this technique, they discovered that there were gene variants in DLGAP2, which is encoding a synaptic protein. And these were associated with individual differences in working memory decline. So we next wanted to examine the possible mechanism whereby DLGAP2 might be promoting cognitive longevity. And since DLGAP2 is expressed in the dendritic spines of hippocampal neurons, and because spine number and morphology have been implicated in both memory and altered in aging, and also because DLGAP2 knockout mice lose spines, we actually quantified the number and type of spines in CA1 neurons from a subset of these DO mice that were randomly sampled from the 18-month-old cohort following their working memory tests. And what we found is that there's a significant increase in the number of thin spines and a decrease in the number of stubby spines in the hippocampus that was indicative of resilient aged mice. Now, notably, these dendritic spine profiles paralleled those that were reported in human AD resilient cases by the Hershkowitz lab years prior. They just didn't know what genes were involved. So next, in collaboration with Tim Homan and Nick Seyfried, we cross-checked our discovery of DLGAP2 against human genetic and omics data from postmortem tissue, and we found a significant association with AD across multiple scales, be it DNA, RNA, and protein, that all supported the idea that DLGAP2 is a resilience factor, that having more of it is better. And so I think the big takeaway of this study, which you can read more about here, is that our studies of normal, non-pathological cognitive aging in mice have real potential for nominating new human-relevant AD targets. And they also provide a system where we can conduct rigorous testing through genome editing, as well as screen interventions in a way that simply can't be done in humans. So we've also identified several conserved modifiers of normal non-pathological cognitive decline using genetically diverse mouse populations. And we've found that they play a key role in setting the intrinsic excitability of neurons in the hippocampus, as well as modulate synaptic plasticity, which are two leading candidate mechanisms of information storage in the hippocampus that are going to come back later in this talk. Work from my lab also developed the first conceptual framework for identifying mouse models of cognitive reserve or resilience in normal non-pathological cognitive decline, specifically leveraging recombinant inbred mouse populations that I'm going to talk about later, which allow us to resample the same genotype across the lifespan to generate pseudo-longitudinal measures of cognition. And then lastly, additional collaborative work with the Churchill lab and the DO mouse population led to the important observation that diet interventions that promote lifespan either have no impact or damaging effects in cognition in aged mice. And we identified variants in the SLC16A7 gene, which encodes a transporter for metabolites in neurons, as being associated with individual differences in cognitive performance across this DO panel. And although I don't have a lot of time today to talk about all of the work that we've been doing in normal aging today, it is really critical because our work in genetically diverse mouse models of normal non-pathological cognitive decline served as the foundation for our development of new Alzheimer's disease models that would allow us to understand how and to what extent causal AD mutations interact with genetic background to confer individual differences in cognitive decline. So here I'm just going to briefly point out some of the work that we've been doing evaluating resistance to Alzheimer's disease. And resistance to Alzheimer's disease is defined as the brain's ability to avoid classic neuropathological changes in the brain, despite risk factors such as predisposition to Alzheimer's disease. And our work in mice and recent work by Patricia Boyle in humans suggests that amyloid burden is not well correlated with cognitive decline, and that it actually explains only a small fraction of the variance in cognitive outcomes in mouse and human populations. And this might be one reason why anti-amyloid interventions have yet to be universally accepted as successful in clinical trials. With regards to examining brain resilience to Alzheimer's disease, this is defined by the brain's ability to cope with those accumulating neuropathological changes in the brain, like amyloid and tau, by actually preventing neurodegeneration. And so Bree Gurdon and my lab applied an atlas-based quantification of labeled neurons in glia across the mouse brains. And to date, we've found no correlation between neuron load and cognitive outcomes in the 6180 mice that she's analyzed so far. And so she's in the process of continuing with a set of 350, and I'm excited to update you all in the next year. But basically what I'm leading up to is the basis of my talk so far, which is that all of the rest of my talk is really going to be about cognitive resilience to Alzheimer's disease. And I'll just point you to our recent collaborative publication with Tim Homan and the AD Resilience Working Group, showing you that the genetic architecture of cognitive resilience to Alzheimer's disease is distinct from clinical AD risk. And what that means is that there are resilience genes that exist and are ripe for discovery. So what is cognitive resilience to AD? So we define individuals exhibiting cognitive resilience as those who evade cognitive decline that's predicted based on the inheritance of deleterious autosomal dominant mutations that are known to cause Alzheimer's disease and are corresponding to very high levels of brain pathology. And in cases of familial Alzheimer's disease that result from mutations in amyloid precursor protein and presenilin 1, a delay in the predicted age at symptom onset can be derived based on family history of carriers and non-carriers. So if your mother's age at symptom onset is 45, and her mother's age at onset was 43, and you're a cognitively normal AD mutation carrier at 60, you would absolutely be considered a resilient individual. In the broader population, we end up having to use residuals of predicted cognitive outcome based on biomarkers, like an amyloid PET scan. And that allows us to identify and quantify the resiliency of any given individual. And because resilience to Alzheimer's disease is really difficult to study in human populations, when I started my lab in 2012, we were studying the impact of human familial Alzheimer's disease mutation on cognition in genetically identical mice to try and identify the molecular and cellular correlates of resiliency. And while there is variable susceptibility within a single inbred strain, can provide some mechanistic insight into cognitive resilience, utilizing only one strain is similar to studying Alzheimer's disease in a single human. And as such, the insights gained are unlikely to generalize to other mouse strains, let alone humans. They're also not addressing the role of genetics, which we know from human studies explains about 50 to 80% of the variation in cognitive decline. And so we developed the first Alzheimer's disease mouse genetic reference panel to better model the heterogeneity of human Alzheimer's disease, and also to identify the genetic mechanisms that are underlying individual differences in susceptibility, as well as resilience to Alzheimer's disease cognitive decline. So we started with some pretty high-level questions, and the first was, does the introduction of human AD mutations into genetically diverse mice yield new insights into human-relevant resilience factors and therapeutic targets? And I'm going to show you two examples where naturally occurring mouse variation can be used to identify translationally relevant memory tasks in mice, and how mapping studies revealed a novel ApoE variant that's associated with cognitive resilience. So here's the model that we generated to identify modifiers of human familial AD mutations on cognition. We bred 5X FAD female transgenic mice that have 3 mutations in human amyloid precursor protein and 2 in presenilin 1, with males from 28 different BXD mouse strains. So the BXDs are a large family of diverse inbred strains that were derived from an initial crossing between 2 common inbred strains that are distant in terms of their genetics. So the C57 black 6J mouse, which I'll refer to as the D6, and the DBA2J strain, which I'll refer to as the D2. And these strains are segregating 5 million polymorphisms across the population. The progeny are a set of F1 AD-BXD strains, each of which contain the same high-risk human mutations, but whose genetic background is different. And this allows us, for the first time, to understand the contribution of individual genetic variation on Alzheimer's disease susceptibility and resilience in the mouse. We also generated the littermate non-transgenic controls that provide insight into the processes of normal, non-pathological cognitive decline. And since each parent strain is inbred, these F1 mice are fully reproducible and can be studied across time and laboratories. So to characterize these mice, we measured their body weight and working memory longitudinally And then at either 6 or 14 months, the mice went through a more detailed phenotyping battery that included tests of sensory motor function, anxiety on the elevated plus maze, as well as contextual fear acquisition and memory testing, followed by harvest and tissue biobanking. So to determine the age at onset of memory deficits in our mice, we chose to measure working memory longitudinally because it depends on the integrity of the hippocampus, which we know is one of the earliest brain regions that's affected by Alzheimer's disease, and because there's little carryover in this task. The average age at onset of working memory deficits is defined at the age each strain is performing at chance, and that's calculated and shown here. So as you can see, the age at onset of working memory deficits varies quite a bit, with some early onset strains that show deficits at 4 months of age, some strains that maintain working memory out until 10 months of age, and those that actually can perform this task out to the latest time point that we tested them. And these would be our most resilient mice. The other important observation was this variation in age at onset across our AD-BXD population looks a lot like the variation that we see in human patients that are carrying familial Alzheimer's disease in amyloid precursor protein and presenilin 1. So at least at this level, they looked like they were a good model for the diversity or heterogeneity of familial Alzheimer's disease. So next, we wanted to ask if the AD-BXD population is a good model of individual differences in sporadic late onset disease, because the genetics of sporadic late onset Alzheimer's disease are far more complex. And so we took advantage of recent studies showing that an individual's genotype across GWAS loci provides an improved estimation of their risk of developing Alzheimer's disease. And this is beyond ApoE. And then we were the first to extend this idea to mice. So we calculated a genetic risk score based on 21 AD risk loci that have variance across our AD-BXD population. We stratified those strains into impaired and unimpaired groups based on their 6-month-old cognitive function. And then we identified the risk allele for each one of those 21 loci. We were then able to use this to determine the odds ratio that each allele pushed a strain towards cognitive impairment. And from this, we calculated a genetic risk score for each strain. And we found that the genetic risk score was significantly associated with late disease cognitive performance as measured in a separate cohort of mice at 14 months of age using contextual fear acquisition. But that was not the case for the non-transgenic BXD littermate controls. And what this told us is that amyloid is sensitizing the effects of your genetic risk, but it's not playing a role necessarily in normal non-pathological cognitive decline. The other exciting thing about this data was it suggested that we could use contextual fear acquisition as a translationally relevant cognitive assay for preclinical studies, which had been a really big barrier in terms of trying to get from mouse to human and improve the translation. So next we performed genetic mapping of this trait, and we identified a single QTL on chromosome 7 that was explaining 21% of the phenotypic variation. The D2 allele was defined as the risk genotype, and our QTL was syntenic to the ApoE locus in humans. So given that there was a known coding variant in the receptor binding domain of ApoE, shown here, we stratified these strains based on their ApoE genotype and plotted their cognitive performance. And we found that the cognitive performance tracked with ApoE genotype, where heterozygous D2 carriers performed more poorly than those that were homozygous B6 carriers. We worked together with Rick Mazer to use CRISPR genetic engineering and develop a B6 mouse with that D2 ApoE allele. And then we crossed that mouse with a 5xFAD heterozygous mice in order to generate 4 groups, carriers and non-carriers for 5xFAD, and carriers and non-carriers for that ApoE knock-in. And then we evaluated contextual fear acquisition performance at 14 months of age in all 4 of these groups. And what we found is that 5xFAD mice with a single copy of that ApoE D2 allele were more susceptible to cognitive decline compared to those 5X-FAD mice that were on a B6 background. So of note, our genetic mapping suggested that 2 copies of the B6 allele in the receptor binding region for ApoE could confer some resilience to familial Alzheimer's disease mutations as early as 2017. And this region was rediscovered as a resilience region in a woman who is a carrier for presenilin-1 mutations and homozygous for the ApoE Christchurch mutations that are located in this exact same region. And so we're now running the molecular dynamic simulations with Zach Levine to determine if the mouse ApoE B6 isoform looks like the human ApoE isoform with the Christchurch mutations in it. And I'm hoping to have those results soon, hoping before AAIC at least. Yep.
Just for those of us who are not ApoE fanatics, the Christchurch mutation is a resilience mutation?
Yeah, sorry. When you're homozygous, so there's one woman that was identified as resilient. She carried 80 mutations and she had 2 copies of the Christchurch. So 1 copy isn't enough, just like 1 copy of the B6 is not enough. That's a great question. Thank you. All right, so given that there are a variety of mechanisms whereby ApoE genotype might confer resilience to Alzheimer's disease, we took an unbiased approach to identify cell types and gene expression profiles in the hippocampus using single nuclear RNA sequencing. And here I'm showing you that the vast majority of differentially expressed genes from 5X-FAD mice with and without that single copy of the ApoE2 allele are in excitatory neurons of the hippocampus, and actually mostly in the dentate gyrus and the CA1 region. Our pathway analysis suggested that neurogenesis, synapse organization, metabolic function, and neurotransmitter transport were all altered by that 5X-FAD ApoE knock-in in the mouse. And so I want to just say that I think the reason that this is so important is it's, to my knowledge, the first demonstration of a role for ApoE in promoting resilience to familial AD mutations through its effects in excitatory neurons. And this differs from published roles for ApoE in glial cells. And I think it just adds to the possible role where ApoE gene can impact cognitive decline and resilience to Alzheimer's disease, which I think is really wild. So I'll just quickly summarize this part in saying that, as many of us appreciate already, genetic background matters. It provides more translational models of human disease, and it also provides the tools needed to identify and validate resilience factors and their mechanisms of action. And I think this is going to open up a brand new area of interventional strategies, which I refer to as resilience-based therapeutics. So next, we wanted to ask, outside of ApoE, where does risk and resilience to these Alzheimer's disease mutations originate in the mouse at the molecular, cellular, and anatomical levels? And I'm just going to quickly tell you before I show you that our data point to specific hippocampal and cortical excitatory neuron types that seem to remodel in response to microglial activation and proliferation. And so first, Sarah Hewer, when she was a graduate student in my lab, was keen to use the behavior we had generated at 6 and 14 months in order to generate decline scores for each AD-BXD strain, and then integrate these data with gene expression and genetic data across the AD-BXD population with the goal of nominating cell types and pathways that best explain individual differences in the cognitive outcomes of these mice, and then cross-check them against humans. And so she took RNA sequencing data from the hippocampus and built gene co-expression modules using the consensus clustering algorithm SpeakEasy. And she identified 31 discrete transcriptional modules, which were all shown here. And this heat map is basically showing you how the modules relate to each other based on their overall gene expression. The second one in gray shows the enriched cell types from our deconvolution analysis using the Ben-Bar's single cell RNA sequencing data set. And she also performed gene go enrichment on the genes within each module to get an idea of the biological processes and pathways that were represented. And together, these modules covered transcriptional networks that were enriched for many disease-associated cell types and biological functions. And so of those 31 modules, 16 of them were significantly correlated with at least one of the cognitive measures that we had made after correction for multiple comparisons testing. And to identify modules explaining the most variance, she performed a linear regression. And as you can see here, there's a number of gene modules whose differences in expression explain a fair amount of the variance in each cognitive measure. Those in red are increased in glia and mostly microglia from susceptible strains, whereas those in blue are increased in neurons from resilient strains. And so these results told us that in addition to the microglia that Gareth talked about last week, we also need to focus on neurons in order to identify resilience factors. But we didn't know exactly which neurons in the hippocampus we needed to focus on. So given that the hippocampus is comprised of a large number of neuron classes and subtypes, we thought to use single nuclear sequencing data to try and link co-expressed resilience genes to their cell type of origin. And to identify cell type specific transcriptional signatures that are associated with cognitive resilience, we performed single nuclear RNA sequencing on hippocampal samples that were collected from 12 AD-BXD strains and their non-transgenic counterparts, where 6 of them were defined as susceptible and 6 of them were defined as resilient relative to the population mean. The nuclei were isolated using manual dissociation and loaded for capture using V3 chemistry and ultimately sequenced. And so gene expression profiles were ultimately analyzed from 220,000 nuclei across these samples, and then they were clustered and visualized here. Cell type identification for each cluster was determined based on overrepresentation of cluster markers from Hippo-seq data, which is a data set that a lot of people in the field use to call cell types in hippocampus. And what I'm showing you here is that all of the major cell types in the hippocampus were represented across our 32 unique clusters. When we subcluster the excitatory neuron class, it revealed that there were 21 cell type clusters that reflected the neuronal diversity that's expected in the hippocampus. And one of the first observations that we made when we looked at differentially expressed genes from susceptible AD carriers to their non-transgenic counterparts are these robust changes in gene expression in the dorsal dentate gyrus and CA1 neurons of the hippocampus, which were for the most part absent in our resilient strains at both 6 and 14 months of age. And so this led us to speculate that those AD-induced changes underlying cognitive decline were somehow being buffered by the genetic backgrounds that were conferring resilience to the AD mutations. When we specifically focused on differential expression of genes in the excitatory neurons from susceptible versus resilient AD mutation carriers, we found that the top pathway is ribosomal protein synthesis, which is really interesting because the candidate mechanisms of information storage that I spoke to you about earlier, intrinsic neuronal excitability and synaptic plasticity, are known to be protein synthesis dependent. And so this data actually provided a really strong premise for our new R01 aimed at using MET-RS and TurboID mouse models to identify newly synthesized proteins in a cell type specific fashion in AD resilient mice, with the goal of identifying novel resilience-based targets. Lastly, we integrated the transcriptional profiles of glial cells, which we know are involved in neuroinflammatory responses to amyloid, with neurons into NicheNet. So NicheNet is a method that allows us to predict ligands from astrocytes, neurons, and microglia that are acting on their downstream targets. In our case, we're looking at those resilient dentate gyrus neurons as well as those resilient CA1 neurons. And so this method ends up combining the cell expression data with prior knowledge on ligand receptor pairs, as well as signaling and gene regulatory networks. And so I'm just showing you here the top ligands that we predicted will promote cognitive resilience to those Alzheimer's disease mutations through their influence on dentate gyrus and CA1 neurons. And I wish I had more time to discuss these with you all today, but to suffice it to say, our results provide a short list of potential secreted ligands that we wanted to test directly on the consequences on dentate gyrus and CA1 cells. And so to do this, we established patch sequencing in the lab with tons of help from the O'Connell lab. Patch sequencing is a modification of whole cell patch clamp recordings that enables mRNA sequencing of the cell contents after electrophysiological recordings from individual neurons, as well as getting information about their morphology. And so this is going to allow us to bath apply those ligands of interest that I showed you on the last slide and evaluate their effects on neuronal function, instruction, as well as their consequences on gene expression. And so this is just proof of concept data showing the functional readout of eight neurons from the hundreds that we've recorded so far, and also the expression of tens of thousands of transcripts and nearly 8,000 genes that we can sequence from a single nucleated patch using smart sequencing. And so to just summarize this next section, we think that these are providing new insights that are pointing us now for the first time to specific neuron pathways, to specific neuron types and their pathways that are associated with cognitive resilience. And given that they're likely influenced by glial cells from Sarah Hewer's work and others, we're now positioned to test that interaction using state-of-the-art patch sequencing and other techniques. Next, we wanted to determine the extent to which resilience signatures are conserved in humans and prioritize candidates that might have a likely therapeutic value. And so to do this, we generated RNA sequencing data from mouse brain tissue that's containing the prefrontal cortex. And we targeted the prefrontal cortex for two reasons. The first is that hippocampus-dependent memories are transferred to this region for long-term storage. And this is a process that we know is disrupted in Alzheimer's disease. And also because we had comparable data from susceptible and resilient AD human cases that had been generated by our collaborators from postmortem tissue. And so in order to examine the alignment between the mouse and human single nuclear data sets, raw data from the human and mouse samples were integrated and analyzed using the package Liger, which was developed by the McCosco lab. And I'm showing you here that after integration of the data and two-dimensional visualization, you can see that the human and the mouse nuclei overlap in terms of the organization into cell clusters. These clusters comprise of all of the major cell types in the prefrontal cortex, the vast majority of which had a cross-species alignment score of 0.65 or greater, and I've bolded those here for you. And these are indicative of well-conserved cell types. So next, comparison of differentially expressed genes from our susceptible and resilient humans and mice pointed us towards cluster 20. Which had the most upregulated genes in resilient individuals, being both mice and humans. And I just want to remind you that the way that I define resilience factors theoretically is that more of it is better. So that's the reason we wanted to focus on differentially expressed genes. So using metaneighbor analysis and cell census network data from the Gillis lab and the Brain Initiative, we were able to identify cluster 20 as layer 4-5 isocortical neurons. And then our pathway analysis revealed a likely role for mRNA metabolism, CNS development, neurogenesis comes up again, as well as viral processes and protein localization, with the overlap of several genes in more than one functional pathway. So among those upregulated resilience genes from cluster 20, a total of 12 of those genes, 11 from the mouse and 1 from the human, had previously been nominated as Alzheimer's disease targets on Agora. So Agora is this interactive web-based tool that was developed by Sage BioNetworks in collaboration with members of the AMP-AD academic and industry teams that include Model AD, Resilience AD, and Treat AD. And so the thing that's important about Agora is that it hosts extensive druggability information that was developed by our industry partners. And so of those Agora nominated targets, only 1 had a good druggability profile. In addition, we identified a total of 61 new targets, which had not previously been nominated, but that we subsequently validated using human proteomics data from the mmary team. And I'm showing you here that of those 61 new targets, only a small fraction are predicted to be druggable and safe. And I think this really drives home the point that we need to continue to search for new gene targets and mechanisms of cognitive resilience if we're actually going to end up with a target that's druggable and safe. So I think our work is really the first to directly test the alignment between the mouse and human prefrontal cortical cells in a cell type specific fashion. And we found that the vast majority of them are well conserved. And I think this tells us that the genetic and phenotypic variation in mice are key to modeling the human disease, and that prior criticisms of the poor translational value of AD mouse models had far less to do with species and had much more to do with the major limitations of studying disease on a single genetic background. We also nominate layer 4-5 cortical cells as key players in cognitive resilience to Alzheimer's disease and propose several new gene targets that might promote resilience with good druggability and safety profiles. These are targets that we're really interested in pursuing in the future. So next, I want to show you how we're moving from a single gene target to the discovery of small molecules with good selectivity and safety profiles. And so as some of you may remember, in 2017, Kristen and I identified the non-canonical cation channel TRPC3 as a potential target to treat Alzheimer's disease. So this target was overexpressed in brains of impaired aged and AD mice, as well as in AD postmortem tissue. Using viral-based gene therapy tools, we showed that a 20% knockdown in the TRPC3 protein was sufficient to prevent cognitive decline in aging mice, and I'm also showing you that here in AD models. But a selective and safe antagonist was not available. Now, as academic labs, we weren't really positioned to run a drug discovery program around this target, but with the help of BJ Borman and Ken Fassman and Annalisa Lucido, we were able to partner with medchem experts in pharma, and this was also thanks to generous funding from JAX and several angel investors. So that's been really exciting, but the thing that I take away from it, being involved in it for so many years now, is that the discovery of TRPC3 came out of years and years and years of evaluating cognition in large cohorts of aging in AD mice in vivo, which is incredibly slow and expensive. So to accelerate the unbiased discovery of new resilience factors, we wanted to develop a 3D co-culture system that exploits the established differences I've shown you in the onset and progression of cognitive symptoms in our AD-BXD panel so that we could establish that as a therapeutic screen. And so with new funding from the Alzheimer's Disease Association and help from the Kaplan Lab at Tufts, Maria and I are now developing a 3D model of cognition to perform unbiased screen of compounds using sophisticated readouts of resilience that are now going to include things like measures of differentiation and neurogenesis that I spoke to you about, cell morphology in terms of the spine number and type, as well as microglial states. And this framework will also allow us to use patch sequencing to evaluate the vulnerability of these co-cultures to things like excitotoxicity, and we especially want to evaluate them in the response to those ligands that were predicted by Neuron to be secreted by neighboring cells and glial cells that influence neuronal function on those dentate gyrus and CA1 neurons. I'll also say thanks to the amazing work by the Reinhold and the Para labs, we already have a roadmap to pursue cellular models of mouse phenotypes, and now we're just going to do it in the context of AD. So last but not least, once we have these targets, how are we going to know what mouse models to put them in? And so in terms of picking AD-BXD strains for drug testing, we wanted to know how well do the hippocampal gene expression profiles align with those that have been recently published and defined as the 3 human AD profiles. And this work was done by Bin Zhang. And so when they took hippocampal RNA sequencing data from postmortem tissue and did unbiased clustering, what they found was there's actually 3 molecular subtypes of human AD. So the first is subtype A. It stands for atypical. It's enriched for synaptic genes. This B type has more of a tau neurofibrillary tangle flavor to it. And then here is this subtype C, which is really a classical amyloid immune response signature. And so Vivek Philip, using the RNA sequencing data in the hippocampus from our 14-month-old AD-BXD mice, applied supervised clustering approach and actually found that there's really high alignment between the molecular profiles of mouse and human subtypes in each of these 3 main clusters. And so what he's showing you here is that, here's where human A falls, human C, human B. So what we're going to end up doing is choosing mice choosing mouse strains that are the closest to their human counterpart for drug testing. We're also going to take into account sex. And so I want to show you here is that we actually found that there's an enrichment of female AD-BXD strains that are aligned to the classic amyloid AD human subtype. And this actually parallels the higher incidence of females with AD, especially that are amyloid PET positive. And so this is telling us that sex can push a strain into a different AD subtype, given that these all started out with equal representation of males and females across this group. So our preclinical studies are going to start with the collection of these 3 mouse strains for both males and females of each of the human subtypes. And this is one way that we can extend drug studies in a single strain to multiple models of human disease. We're also going to leverage data from a very large study in over 6,000 mice that tells us the importance of both sex as well as diet and genetic susceptibility to cognitive decline. And that's really critical information when we think about moving forward with cognitive testing. So what I'm showing you here is that even a classic risk factor like high-fat diet is not bad for every strain, and its impact on disease really depends on both genetic background, which is represented here, as well as sex, where we're comparing and contrasting how males and females are responding to a high-fat diet. So basically what I'm telling you is there's a set of strains over here that are actually less susceptible to high-fat diet in terms of their cognitive decline. And these strains are actually much, much, much more susceptible to the effects of high-fat diet. And there are some strains that do better on a high-fat diet, particularly females. And so when we put all of these pieces together, my last slide is really illustrating the current and future work that's being pursued through collaborations with so many of my colleagues here today, for whom I'm incredibly grateful to work with. So in addition to aligning the AD-BXD strains to human AD subtypes, we're also generating mixed pathology models for ADRD with Kat Lutz and Kristen O'Connell, as well as Q111 BXD models of Huntington's that will all be maintained on genetically diverse backgrounds in order to characterize them and test their alignment to the human condition. Our goal here is to define models of susceptibility, to test resilience-based interventions, and evaluate how well they generalize to new models of late onset Alzheimer's disease from the Model AD Consortium, but also other neurodegenerative diseases like Parkinson's and frontal temporal lobe dementia. We're going to continue to integrate phenotype data, but go well beyond cognition. So as an example, we're working towards validation of polygenic risk scores in mice that are aligned to GWAS loci for Huntington's traits. And this is being done in collaboration with Jim Gisella at Harvard as part of our new R01. And we're also doing the same for psychiatric traits in collaboration with Rob Sweet as part of SpeakEasy. The assays that are sensitive to genetic risk for Huntington's, psychiatric, as well as motor and sleep phenotypes with Vivek Kumar and Kristen O'Connell are going to be really powerful readouts for preclinical evaluations of interventions. And we hope that they will help us identify global resilience mechanisms that cut across neurodegenerative diseases, should they exist. We also have new funding to continue to design these in vitro 3D models of mouse AD subtypes to test resilience targets and perform drug discovery screens. And then now we're positioned to test these compounds in relevant AD subtypes with both behavior and MRI, thanks to the CBA, as our readouts for target engagement. And we're going to share this information openly so that our discovery efforts and our strategies can be evaluated by AMP-AD as well as the greater community. And so with that, I want to thank you all for your attention. I want to also thank the JAX Angel investors who've been advising and supporting the TRPC3 drug discovery program, which is really my first attempt at bridging the gap between basic science discoveries and therapeutics. And so it's been very challenging, but also incredibly fun. And I've learned a lot from Kristen and BJ and others. I also want to thank Tony Evnen for his generous support, and also because he's really generous with sharing his deep network of academic and industry contacts that have come in really helpful. I want to thank my entire lab, past and present, and I want to give a special thanks to Kathleen Silva, Tim Leach, Amanda Adams, and Kat Widmeyer for keeping all of our aging studies going during the pandemic. We really, really treasure you. These guys are so much fun. I want to thank Alan and the scientific services. I should have put that slide last, sorry guys. I'm going to thank Alan and the scientific services, and a huge shout out to the CBA, especially Laura Anderson, and single cell biology and protein sciences. And then Annalisa and Tonelli have had a hand in all of our successful grant funding and none of our failures. And so if that doesn't tell you something about who you should be talking to, as much as I really want them to keep working with me, I really encourage everyone to try to get into that group. That's a really elite group. I want to thank Nadia and the leadership at the lab. They've created opportunities that every scientist dreams of. This is a really special place. Having been in other institutions, there's nothing like it. My external collaborators that make the mouse to human translation possible and fun. My faculty mentors, Gareth and Alyssa, and all of my collaborators, and of course, all of the JAX faculty. Our funding, philanthropic, NIH, foundations, again, Annalisa and Tonelli, their names will come up again and again. And last but not least, I want to thank my amazing family. This is my dog, Huxley. He thought he wanted to watch my talk, and this was what he looked like afterwards. So not his favorite thing. And then also, I know a lot of you guys don't know my daughter, Caroline, but she's been doing science with me since I was an undergrad. And she's all grown up now and smarter than me. But anyway, if you ever get the chance to meet her, she's a real hoot. And with that, I'm going to change this slide and I'll take questions.